Okay, so this is going to be the last lecture of the semester. So just a few reminders before we get into the material. Extra credit opportunities. There's still the online survey. If you haven't filled it out yet, please do so. That is for extra credit. I can only see who took it. I don't see the results. And the course evals, last check, almost at 90%. So please make sure you finish those up before those close before finals. So if you haven't finished your course evals yet, you'll want to do it. Learning objectives for the final have been updated on eClass. The last few learning objectives will be updated after this is posted because they may change depending on how far we get. The final homework questions are updated on eClass for Chapter 20 with the population genetics. Final exam is Thursday, 8 a.m. It is in the regular classroom. Review sessions. Sunday's review session is going to be moved back just a bit, 1:30 to 3:30. Monday, I'm going to do 2 review sessions because I have a meeting from 2 to 3, so I'll do one from 1 to 2 and another one from 3 to 4 in the genetics lab if people are interested. Tuesday, I'm going to do a review session in the view. I'm going to start at about 9 o'clock, and it'll go until people run out of questions. And then Wednesday from about 2 to 4 in the genetics lab. I'll be around most other times, so if you need to meet, just let me know. Also a reminder, exam keys are available in the science office this week. So make sure that you sign in when you go ahead and you look at the exam keys, and you'll want to go and make sure you're looking at the answers to the questions. This week I'm available by email. You can, if you want to Skype, let me know, just shoot me an email and we can do so. Also, please turn in your lab notebooks next week. So Tuesday and Friday, you turn them in on Monday. Thursday, depends on what Dr. Penna told you. Same thing with lab reports, so make sure that you get everything in before the end of the semester. All right, so we're going to finish up Chapter 21 today. We're going to continue talking about the Hardy-Weinberg principle and how to use it. And then we're going to talk about some of the examples of when populations are not in Hardy-Weinberg equilibrium, such as genetic drift, mutations, natural selection, and things like that. So what did we do last class? So we used the Hardy-Weinberg principle to determine if a population was in Hardy-Weinberg equilibrium. And these were the steps that we did here. So calculated the allele frequencies, computed the proportions for the alleles, calculated the expected number of alleles, and then performed the chi-square test. So we did this with our these numbers here. So if you remember, we got a p-value of 0.33. So we did not reject the null hypothesis. Our null hypothesis is that the population is in Hardy-Weinberg equilibrium. Since these numbers are not significantly different, we would say that this data is consistent with the population being in Hardy-Weinberg equilibrium. So I asked you to look at this population, the Morton Island one, before you went through this online lecture. So what about this population? So these are our genotypic frequencies. Do we think this population is in Hardy-Weinberg equilibrium? Well, we do the same thing. We calculate the expected number based upon first calculating the allele frequencies. And I'm not going to work through this. You can calculate this. It's the exact same way we did it with this. But you're going to calculate the allele frequencies. And then based upon that, you're going to calculate the expected number of the red, the gray, and the white kangaroos. We're going to compare them to our observed number and simply do our chi-square test here. And we get a chi-squared value of 247.6. Remember, the degrees of freedom are 1 because even though there are 3 phenotypes, we're actually measuring alleles here. We're interested in, are the allele frequencies in Hardy-Weinberg equilibrium, and there are only 2 alleles. So 247.6 would be way off the chart here. This p-value is practically 0. So what does this mean? It means that this population is not in Hardy-Weinberg equilibrium here. So the first population was an example of it being in Hardy-Weinberg equilibrium. This population is an example of it not being in Hardy-Weinberg equilibrium. What does that mean? Well, we'll talk about in just a few minutes what it means. What are the things that oftentimes apply when a population is not in equilibrium? But a few more things about Hardy-Weinberg. So we can also use this law to to calculate the frequency of heterozygotes in a population. This only works if the population is in Hardy-Weinberg equilibrium. So if we know that the Hardy-Weinberg population is in equilibrium, then we can calculate the frequencies of all three genotypes if the frequency of either allele is known. So what does this mean? Well, let's say you're done with kangaroos. Let's work on koala bears now. We're going to research koala bears. Well, you count all the koala bears on the island of Tasmania, and you count 881 of them, and you find 17 albino. You know that this is a recessive trait, albinism, similar to humans, where you need two recessive alleles. If you assume that this population is in Hardy-Weinberg equilibrium, you can calculate the allele frequency of the carriers of this allele, which is oftentimes what we're interested in doing in the population. We'll talk about why in just a second. So how would this work? Well, if you know that 17 out of 881 were albino, those are going to be little a. And remember, so little a is the same as, so that the recessive allele is the same as Q. So little a is is a squared or Q squared. So the frequency of Q squared, the homozygous recessive, is 0.019. Well, if this population is in Hardy-Weinberg equilibrium, then P squared, or I'm sorry, if we take the square root of Q squared, so the square root of 0.019 is 0.139, that's going to give us Q. So that is the frequency of this allele in the population, 0.139. We know that P has to be 1 minus 0.139 because in the Hardy-Weinberg equilibrium, both alleles add up to 1. So P, this would, or we'll call this big A, would be 0.861. Q, or little a, would be 0.139. Well, what is in the Hardy-Weinberg equilibrium? Heterozygotes are 2pq. So 0.139 times 0.861 times 2 is going to give us the number for 2pq. So this is what this is giving us here is this is our homozygous recessive genotypic frequency. So this is the frequency of this genotype, homozygous recessive, in the population. This is the heterozygote genotypic frequency. So we would expect that about almost 25% of the population would be heterozygous, even though this particular trait is not very common. So again, if you know one of the genotypic frequencies, you can calculate what the allele frequency is, and then you can determine what the frequency of the carriers are in the population. So let's review the two types of calculations we've talked about so far. The first is we use to determine if a population is in Hardy-Weinberg equilibrium. And to do that, we calculated the allele frequencies. We calculated the Hardy-Weinberg proportions. So these would be the these would be the P and Q, so the alleles. We determined the expected genotypic ratios from those Hardy-Weinberg proportions, so based upon the total number of organisms in our population. That's where you multiply the expected proportion times the total number of organisms, and then you perform chi-square tests. So we did this one in class, and we determined that the population was in equilibrium. So this is using sample data to determine if the population is in Hardy-Weinberg equilibrium. What we just talked about was if we wanted to determine, to estimate allele frequencies, using the Hardy-Weinberg law to do that. So you can calculate the frequency of, for example, homozygous recessive. So that would be our albino here. That would be our Q squared. Then you can determine the allele frequency because that's going to be the square root of Q squared, or Q. You determine the other allele frequency, which is going to be 1 minus Q. And then you can calculate the frequency of the heterozygotes, which is 2 times P times Q. But keep in mind, when you do this, you're assuming the Hardy-Weinberg equilibrium is in effect. You can only do this if you are operating under the assumption that the population is in Hardy-Weinberg equilibrium. Otherwise, you cannot do the second calculation. So again, just to a reminder, when you know the genotypic frequency here of, say, this is an example of homozygous recessive, usually that's what we can see, you can determine the genotypic frequency, you can determine the allele frequency, and then you can ultimately determine the frequency of heterozygotes. And what you see here is this is a pretty rare trait, or a pretty rare allele. So only 0.02, so about 2% of the population show this recessive trait. But almost a quarter of the population are carriers. They're heterozygotes. And this is very common among rare alleles in the population. So what does this mean? Well, if in terms of implications of the Hardy-Weinberg principle, one, we've talked about number one so far. Allele frequencies remain constant from generation to generation. So if a population is in Hardy-Weinberg equilibrium, the frequencies of the allele are not going to change over generation to generation. However, also, if you have a rare allele in the population, the frequency of the heterozygotes is going to be much higher than the homozygous, which is very rare. So we just used the example of koalas, but let's look at another example for this. So let's say that the frequency of the rare allele is 0.1. So what does that mean? Well, if you do a little math for the Hardy-Weinberg equilibrium here, and this is just some cross multiplication, you're looking at the ratio of heterozygotes, 2pq to q squared, and you're just crossing out a q here, 2p divided by q. This math shows that the heterozygotes here are going to be 20 times more common than the homozygotes. So based upon the allele frequency of being 0.1, whenever the allele frequency of something is 0.1, the heterozygotes are going to be 20 times more common than homozygotes. And this is relevant when it comes to certain diseases that are present in the human population. A good example of this is cystic fibrosis. It's very rare. It only affects 1 in 1,700 newborns. So if you divide 1 by 1,700, that's 0.0059. That's q squared. That is because it's a recessive trait. So 1 out of 1,700 people are going to be little a, or so that's our q squared. If you take the square root of that, that's 0.024. So that's the allele frequency of the recessive allele in the population. So p plus q has to equal 1. So this should actually say p here. P is going to be 0.976. This is a misprint. So 1 p plus q equals 1. So p equals 0.976. So what does that mean? Well, remember heterozygotes in the Hardy-Weinberg equation are 2pq. So 2 times p times q here is going to be 2 times 0.976, which is p, times 0.024, which is q, which is 0.047, which actually comes out to a fraction of 1 out of 21. So even though only 1 out of 1,700 people are affected by this disease, or 1 out of 1,700 newborns, 1 out of every 21 people carry it. And this is why if you have, oftentimes if you have a child, if you're pregnant with a child, they will test the parents for this particular allele because it is relatively common. Now the odds of both parents having it is 1 over 21 times 1 over 21, which is pretty small. And that said, this frequency changes. This frequency is different in different populations in the world. So it's much higher in Caucasians and Ashkenazi Jews. Very low. It's much lower in Asians. So, and this has to do with different things that we'll talk about in a few minutes when there are aspects of the Hardy-Weinberg equilibrium that don't apply. So this is why it's important to understand this the fact that a rare allele is still very common in the population in heterozygotes. This is how rare alleles can persist in the population. And this is why, again, they test for it. So this is a good point. If you want to take a break, go back, take a look at the calculations we've done. What we're going to want to think about now is, so everything we've talked about before is testing to see if Hardy-Weinberg equilibrium, if a population is in Hardy-Weinberg equilibrium, or assuming a population is in Hardy-Weinberg equilibrium and then determining the allele frequency from that. But most populations are not in Hardy-Weinberg equilibrium. It's actually quite rare. And if they're not met, if these assumptions are not met, then the gene pool changes. And this is where it's of interest to population geneticists. Oftentimes they start out with a baseline that a population is in Hardy-Weinberg equilibrium, knowing that usually this is not the case. If they do the test and they find out the population in fact is not in Hardy-Weinberg equilibrium, then they're going to look at which of these are going on, which of these are occurring that's going to result in the population not being in equilibrium. So an example, let's look at the example here. They talk about in your book, the frequency of these different alleles for eye color. And you can see P is the dominant allele, A, which is the brown allele, and Q is the recessive allele, G, which is the blue allele. And you can see here that in certain parts of the world, like Africa and East Asia, the P allele, or the A allele, the brown one, is much higher frequency than in parts of, say, Northern Europe, where the G allele is in a much higher frequency. So you might want to think about what's the reason that we have these different frequencies in different parts of the world. And like I mentioned, the Hardy-Weinberg equilibrium, it's a starting point for modeling how populations change. Most of these are not met in natural populations. So this equation is useful for estimating how the population might change if these don't apply. I mentioned in the book they do go over a lot of the math of this. Because we don't have time, I don't expect you to understand the math behind these changes. What I'm more interested in from this point on is for you to understand the concepts of why these affect populations. I'm not going to ask you to do any math about these 5 things. I just want you to understand the concepts for why these affect the alleles in the population. So let's start with one of them. There's 5. We're going to start by talking about genetic drift. So genetic drift is when you get random changes in the frequencies of alleles, and these are usually neutral alleles, due to what is oftentimes called accidents of sampling. And what we mean by this is it has to do with the population size. So accidents of sampling are when there are changes in the allele frequency in populations that are due to small population sizes. Large population sizes are relatively immune from genetic drift. It's really the smaller population sizes that you get these what are called accidents of sampling. Well, what does this mean? Let's look at what this means when changes in allele frequency occur due to small population sizes. Well, again, just a little more background. The smaller the population, the less genetic variety it has, and that makes sense. Larger populations have greater genetic variety. The smaller it is, the less variety on the whole they're going to have. In a very small population, because of this, alleles can be lost over one generation or a few generations simply by random chance. And this is a key part of genetic drift. Genetic drift is random chance. So we'll talk about another aspect of deviations from Hardy-Weinberg when we talk about natural selection. Natural selection is not the same as genetic drift. Genetic drift has nothing to do with any selective pressure. It is just a matter of math. It is random chance that you lose or fix an allele. Because of when this occurs, when a population evolves only because of what this random sampling error, you have genetic drift taking place. In your book, they talk about these Monte Carlo simulations, and they talk about quite a bit of math of it, but with these. But really, what you need to understand about these is the difference between the one on the left and the one on the right. They run the same number of simulations. Each of these, they ran 6 simulations. And what these simulations do is they just look at what are the probability of the different individuals getting matched up in a population. So each of these, you're starting with all heterozygotes. Because the frequency of allele A is 0.5, it means the big A is 0.5, it means the frequency of little a is 0.5. So you're starting with all heterozygotes. In a small population, if you start with all big A, little a, and you randomly match people up, and this is key, randomly match people up, then what's going to happen is some of the time you're going to find the alleles stay about 0.5, and they'll jump up and down a little bit here. So the red and the brown. But some of the time, the big A allele is going to win out, just due to random chance how these individuals match up. Some of the time, and that's what we see with the green and the yellow here. Some of the time, the little a allele is going to win out. That's what we see with the blue and the purple. So when an allele wins out, it gets fixed, which means it's the only allele in the population. When an allele loses, the other allele loses, that allele gets lost. If we look at the one on the right, where we start with, instead of 10 individuals, like we had on the left, we start with 500 individuals, you see, if you run the simulation 6 times, where you randomly match people up, you see much less change in the allele frequency, because you don't have this sampling error. You don't have the random ability of certain alleles to become overrepresented or underrepresented in the population. This is another way of looking at it, similar graphs. So the top graph is population size of 10, and again, starting all heterozygotes. With a population size of 10, by generation 50 or 60, one of those alleles is going to get fixed, and one of them is going to get lost. And it's random which one. You see, half of the time it's one allele, half of the time it's the other allele. Population size 100. By 100 generations, there are changes in the allele frequency. None of them have become fixed here. Population size 1,000, the bottom graph, the allele frequency stays pretty close to 0.5, no matter how many generations you go to. So here what we're looking at is the effective population size, which is the number of individuals that you need for in a population that pass genes to the next generation. So we're not talking about all individuals, we're talking about individuals who pass the genes to the next generation. The smaller the effective population size, so this is an effective population size of 10 up here, the faster the population will drift, which means the faster one allele is going to get fixed and one allele is going to get lost. The larger the population size, the more protection you have against these alleles getting either fixed or lost. So fixation is when there's only one allele left in a population, and all individuals are going to be homozygous. And in the absence of other factors that violate the Hardy-Weinberg equilibrium, like mutations or migration in and out of a population, there's not going to be any more change in the allele frequency in that population. That's why we say it's fixed. It's not going to change. So each of these generations are pretty small, but over many generations you can see large changes in the allele frequencies. So, and you don't have to worry about this number down here, but what's important is to understand the larger the population, the less chance you have of an allele becoming fixed or lost. There are two effects that can greatly influence whether genetic drift occurs or not. One of them is called the founder effect, and one of them is called the population bottleneck. Founder effect is when you're separating a few individuals. from a larger population, you establish a new population. So here, let's say this population, each person has 2 alleles. Some are homozygous blue, some are homozygous yellow, some are heterozygous, and then one person has this red, maybe due to a mutation. And then you pick a very small group of people, and you just randomly end up getting 4 people who are homozygous blue, and then 1 person who is heterozygous with this red. And then as that population spreads out, now you don't have any yellow left down here, and you simply have blue and red because they've established a new population. Population bottlenecks are very similar, but instead it has to do with proportions of individuals dying off. So and this is another way of looking at the founder effect. You have a large originating population. Let's say this was red and green are the 2 alleles, and in a large enough population, those allele frequencies are going to stay about 0.5 each. But if you pull out just a few here, so now we just have a handful of individuals, there's a good chance that one of those alleles is going to get fixed and one of those alleles is going to get lost. And the example here is where the red allele gets fixed by generation 5 and the green allele is lost. A good example of this is in Pennsylvania. So there's what's called the Old Order of Amish in Pennsylvania, where 200 German immigrants started this particular group of this small population. One couple had the allele for polydactyly. It's a recessive allele, which results in 6 fingers and toes. Now, normally, if this was a very rare allele in the population, you would not see many people who would be homozygous for the allele. Because remember, rare alleles have a high proportion of heterozygotes, but low proportion of homozygous recessive. But in a founder effect where you're starting with a much smaller population size, and you also have inbreeding, you have a much higher chance of that allele becoming common in the population. Another example in this population is this type of dwarfism. The allele is very rare in most populations, but it's about, 70 times higher in this particular Amish population because of this founder effect. Bottleneck effect is similar in many ways. You start with a parent population, but only a handful of that population survives. And if that handful has a specific genetic makeup, you're going to see a change over the next generations here, where you end up getting much more of the blue allele than the yellow, even though the parent population was probably about 50-50. So a good example of this is cheetahs. So about 10,000 years ago, cheetah populations crashed due to severe climate change in one of the ice ages. Only cheetahs in Africa and Eurasia survived, and this had to do with locations, their location, not their genes. So you had this small founder population of cheetahs, and because of that, today cheetahs are genetically identical. And this is actually not a good thing because that makes them much more susceptible to certain diseases, where if there was more variation in the population, they'd be much more resistant. We just missed International Cheetah Day. It was yesterday, so feel free to celebrate a day late. I'm going to stop right there for right now, and we're going to break this into 2 separate sections. This will be the end of the first section. The second video will be right next to it on eClass.